Okay, so now we look at these phospholipid molecules. This phospholipid molecule. So when you look at the phospholipid molecule, when we have the head and the tail, so this head we know that they are hydrophilic, and the tail is the hydrophobic. Correct. Okay, so means the hydrophobic means that they are afraid of water, phobia, afraid of water. So when you actually disperse them as a single layer, when dispersed as a single layer, then you realize that they will have these hydrophilic heads that actually interact with water molecule. Can I see that interact water molecule? Means that they will be able to submerge themselves in the water, but because of this non-polar tail or hydrophobic tail, they will project, away from the water. Right? But it doesn't mean that when you are dispersed like this, you'll be able to touch like a fur. Okay, no, because they are very, very tiny molecule. You won't be able to see this. Okay, don't think that, okay, I'm going to put my finger here and then touch it. It feel like a fur. No, you won't get this kind of the feeling. Okay. Now, but if you shake the phospholipids with the water molecule, water, they will form a very stable ball-like structure known as micelle. Now, what's this micelle? If you look at this, because we do know that hydrophilic head and hydrophilic tail. So hydrophilic head, we're going to face the aqueous, means the outside are all water. So inside it form a hydrophobic core. How they form? They form by hydrophobic interaction. Now, always remember when I have hydrophobic tail, so how they interact each other in this case? How they interact each other? They interact through the hydrophobic interaction. So this hydrophobic interaction allowed them to actually interact together and then not join, but how to say that? Place themselves, okay, tier with tier together. But when you shake it out, what will happen here is they tend to form structure like this. Okay, let me draw it out. 2D. And again, can I see that? So water molecule outside, so they can interact with water molecule. Okay, because hydrophilic, right? Interact. Okay, then the tail, all the tail will facing each other by hydrophobic interaction. So outside core, outside hydrophilics, inside the core here will be hydrophobic. Okay, so all hydrophilic head face outwards, forming hydrogen bonds with water molecule. So it means that they're going to interact with water molecule by forming this bond called hydrogen bond. I will tell you guys hydrogen bond more when we enter into the osmosis, okay? And the hydrophobic tail point toward each other, forming this by this hydrophobic interaction. So means that the tail will be shaded by the hydrophilic head. The head here try to protect, something like, okay, protects them. Alternatively, it may form two layers of structure of phospholipid molecule. It forms like a sheet like this, like paper. Okay, so as long as they can actually protect their hydrophobic tail, so the hydrophobic tail won't expose to water, then they will try whatever methods to make sure that this hydrophobic tail won't expose themselves to water. Okay. Now next. But if you shake them vigorously, mixed with water, they will form this structure highlight liposome. Now, liposome is not in our syllabus, okay? But few time question actually come out, okay? And also the structure of liposome in A2, we're going to learn about them again, okay? How liposome will be used in the gene therapy, okay? Now, what this means is, you can see that we say that they may form the two layer like this called bilayer, like a paper sheet. But if you shake them vigorously, they form a liposome, something like a ball-like structure, okay, with the membrane and then interior core. So now it will be something like this. Draw it. So hydrophilic heads. And then the tier interact with each other, a tier. Okay, so if you write it down, the center here again, it will be hydrophilic. Can I see that? Outside also hydrophilics, and the internal core here, will be hydrophobic. So this is a structure of liposome. Okay, structure of liposome. So liposome are microscopic vesicle contain an aqueous core, we call here aqueous, enclosed in the phospholipid bilayer. Unlike micelle, liposome have both aqueous interior and exterior. Okay, so you can see that inside aqueous. Okay, and here, can I see that? It's a hydrophobic, hydrophobic, and the exterior will be hydrophilics again. So liposome is something, we also call it as something like a cell, right? So it's a cell, you have the membrane, and then inside what we have? Cytoplasm. So liposome is basically how do I say that? It mimics the cell, it mimics the cells. Okay, so the type of fatty acid tail of the phospholipid will affect the membrane fluidity. Okay, so fatty acid can vary in number of double bonds. When I say double bond, when you look at this, when we talk about saturated fatty acid, be careful, huh? Saturated fatty acid, they will have maximum number of hydrogen atom in the hydrocarbon chain. So if you look at this, if they have okay, saturated fatty acid, basically they are relatively linear. When they are relatively linear, means that we can pack them very, very nicely. So it will form a very viscous membrane when you pack them nicely, right? You try to imagine if I pack you guys very, very tightly packed. Okay, you stand nicely, okay? Without opening your hand, you stand nicely. So it means that the space for you guys to vibrate will be very, very small, correct? You try to imagine everyone packed like sardine. Everyone packed like this. Okay, each single of you. So can you vibrate? Difficult for you to vibrate, right? So therefore, the membrane tends to be viscous. Are you clear? Clear? Then why is the problem? Is the membrane too viscous? If the membrane too viscous, diffusion can't take place in high rate. Okay, a lot of the transport across the membrane cannot take place. Like for example, exocytosis, endocytosis, diffusion, osmosis, okay? Particularly diffusion won't be able to take place well. Are you clear? But if the fatty acid tail here is an unsaturated fatty acid, when we have the CC double bond or CC triple bonds, then you can see that this CC double bond or carbon-carbon triple bonds will cause us to introduce what we call the king. The king basically means the band. Can you see that? The band. So now they are not packed like sardine anymore. Okay, can you imagine? Okay, we have the band like this. So means that they have space. Now, can you see that they have the space for them to vibrate? So because they have the space for them to vibrate, so means that when they vibrate, they create the gap for the item or particle to diffuse across. Can you see that? Because of this vibration. So this kind of membrane, we call them as a fluid membrane, increase the fluidity. Okay, so always remember unsaturated fatty acid, because they have these CC double bonds or CC triple bonds, so they have fewer hydrogen atom compared to the saturated one, and they introduce a king into the hydrocarbon chain, it will disrupt the packing. Are you clear? It's disrupt the packing. So means that you will be able to see them vibrate well, then diffusion can take place in a higher rate. Okay, allow the diffusion to take place. So it depends on how we pack them. Okay, so at low temperature particularly, because low temperature indicate what? Kinetic energy is low, right? So when kinetic energy is low, then they vibrate less, so the membrane tends to be dense and less fluid. Are you clear? Okay. So a membrane containing a high proportion of hydrophobic unsaturated fatty acid will stay fluid at a low temperature. So my mistake here, guys, this is B, change it. This is A. Okay, so a membrane, okay, with straight tail of the saturated fatty acid packed tightly together, so less fluid membrane. So it's a 2.10B. Okay, diagram. So a membrane containing high proportion of phospholipid with unsaturated fatty acid will stay fluid at lower temperature because they have the space for them to vibrate. So similar to the animal fat compared to the plant oil. right? Animal, we always talk about the fat because solidified. Why? Because they tend to pack very, very nicely, okay? Because of the saturated fatty acid. So one thing here, I want you guys to highlight how you write it. It's very important. We write phospholipid with unsaturated fatty acid. Can you see that? Or saturated fatty acid. You cannot say phospholipids, okay, or saturated or unsaturated phospholipid. Are you clear? Phospholipid cannot be saturated or unsaturated. Only fatty acid can be saturated or unsaturated. So when you write it, okay, means at this moment, your chapter 1, chapter 2, I won't expose you guys to the structure question yet at this moment. Okay, we answer the objective paper first. Okay, so but you cannot run away from this structure question. When you answer structure questions, when you use a term, you must be extra careful, because when I talk about phospholipids, okay, you never say that saturated or unsaturated fatty. I say saturated or unsaturated phospholipids. You only can say phospholipid with unsaturated fatty acid tail. Okay, so many organisms, for example, the fish, they can adjust the physiological to co-environment by increasing the proportion of phospholipid with unsaturated fatty acid in their membrane. So means that if, let's say, at high temperature, higher, not very, very, very high, when I say high temperature, you can see that they're not so viscous, because higher temperature indicates that higher kinetic energy. So they still can vibrate. But the problem is when the temperature is low, co-environment, and then this co-environment, the kinetic energy is low, so therefore you can see that they won't vibrate much. So it will cause the membrane to be viscous and less fluid. So the fish, there's some organisms like bacteria, how they do it? So they exchange. So it means that they take back some of these and then place some of this unsaturated fatty acid tail. So when they do this, then it will make the membrane become less viscous, increase the fluidity. So the organelles that are in charge for this, right now, which organelle in charge of this? Involve lipid synthesis, right? So smooth endoplasmic reticulum. Okay, organelles that are in charge for these regulations, okay, is by smooth endoplasmic reticulum. Okay. So now in activity 2.4, I have some structure of the fatty acid. So which one actually increase the fluidity? Which one decrease the fluidity? Okay, so you compare in this case. Number 1 and number 2, you compare. Okay, number 3 and number 4, you compare. And then number 5 and number 6, you compare. Number 5, number 6, a little bit hard for you guys because you guys haven't done, okay, these are chemistry about these double bonds, okay, what we call the cis and trans positions. Okay, so I will draw it later. So you can try it 2 minutes. Okay, so how you compare, okay? How we compare number 1, and why do we compare number 1, number 2? Because the number of carbon actually the same. Are you clear? The number of carbons actually the same. 1, 2, 3, 4. When I say one zigzag, one corner, actually is 1 carbon. But the difference here is one is no double bond, one we have the double bond. So palmitic acid and palmitoic acid, what make them different? The double bond here, because of double bond, you can see that the structure, palmitic acid relatively linear. It's not linear, but relatively linear, the molecule. But palmitoic acid, you can see that you have the kin, it will bend. Okay, so it bend, it won't pack nicely, right? So means that you're going to increase the fluidity. Are you clear? Increases the fluidity if you use if any organism use palmitoic acid more in their membranes, then you're going to increase the fluidity of the membrane. On opposite hands, palmitic acid actually, if they use it, you're going to decrease, okay, the membrane fluidity. Okay, so number 3 and number 4. What's the difference between number 3 and number 4? Number 3 and number 4, you can see that, yeah, they have one chain here. So one is tuberculosteric acid, another one is a methyl palmitic acid. Both of them, no double bond, but one longer, one shorter. Now, we need to know that if two molecules, if they are large size, the attraction force will be stronger. Are you clear? You learned about this before, right? And two objects, when they are large, so the attraction force will be larger. So that's why iodine tends to be you look at iodine, it tends to be a solid. Bromine, right? It tends to be liquid. But chlorine and fluorine, they tends to be a gas because they're smaller in terms of molecule. Therefore, they tends to have weak interactions. Used to call it as a van der Waals forces, right? Last time. Okay. So in this case, when something's large, long, the attraction force stronger. So when attraction force stronger means that they're going to restrict them from moving. You try to imagine someone hold you well, can you vibrate? No, right? If I hold your hand, hold you well, okay, you won't be able to escape, right? But if I didn't hold you well, then you still can escape, you still can vibrate. So therefore, the longer the hydrocarbons, can you see that the longer the hydrocarbons, then definitely it's going to decrease. Are you clear? Because the strong interaction force makes sure that this long hydrocarbon, they won't be able to vibrate well. Okay, how about methyl palmitic acid? Shorter, if you use this, then definitely can increase because the force, the interaction force between this short chain won't be so great if compared to the tuberculosteric acid, which is longer. Okay, so this is very, very important. You should be able to get it correct. Okay, and then let us look at this cis and trans. Now, what happened in this cis and trans? If I draw out the structure, let you guys have a look. When you talk about cis, basically means that you can see that small molecule at one side, they didn't cross. But for trans, can you see that? The molecule actually in the opposite. Can you see that in the opposite? Now, what will happen here is, you look at this, when you have this carbon, what will happen here is, if a carbon, when you have double bond carbon, then the next carbon will be like this. So that's why it bend. Are you clear? It bend this way. Okay, so means that you have the hydrogens and hydrogen here. So this one is known as cis because the hydrogen and hydrogens actually at the same position. So cis, definitely you can see that the overall structure bend. So we won't be able to pack, we won't be able to pack our phospholipid molecule tightly. But how about the trans? Trans happens like this. Okay, so you have C, 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 double bond C. Okay, because of the H, one H here, another H here, in making the molecule now relatively linear. Can you see that? Are you clear? So in terms of overall shape, this one will actually introduce the kings, but trans actually relatively linear in terms of their shape. Are you clear? They don't bend. So if king, definitely increase the fluidity, but if linear, definitely going to decrease. Okay, the membrane fluidity. Okay, now why I put it this doesn't mean that you do not need to know, because in the objective question come out before talking about the cis and trans, but question won't ask you to label which one is cis, which one is trans. Are you clear? Generally, natural occurring fatty acid, majority of them, they are cis form. So that's why in our oil, we had olive oils, okay, from the palm oils, okay, those are in liquid form. Why? Because they are cis. But in the process of food processing, sometimes because of the high heat, they can convert the cis fatty acid into the trans fatty acid. So you can go to have a look. A lot of the products, for example, you buy the bread, okay, Gardenia bread particularly, they will state that no trans fat, because trans fat is bad. Trans fatty acid actually is bad. Why? Because they are relatively linear, so means that they're going to be solidified at low temperature. They tend to be solidified at low temperature. So with the trans fat is bad for our health. So a lot of the products, they claim that they don't have the trans fat. Are you clear? So what's the difference cis and trans? Cis basically the structure introduce you with the band. But trans, relatively, can you see that they are linear? So when relatively they are linear, means that we can pack them nicely. They are going to decrease the fluidity of the membranes. Clear? So with this, I've done for the phospholipids molecule.